Welcome to the future of understanding. This is NeuroMind Insights, where we unlock the power of your mind.
To sustain life, the human body must continuously break down complex, jagged macromolecules from its environment and convert them into uniform, usable energy. This happens inside a compartmentalized biological refinery. The system must deploy mechanical forces and caustic acids strong enough to annihilate complex organic matter, while simultaneously preventing those exact same tools from dissolving the host's own internal tissues. This process occurs within the lumen, a single unbroken pipeline extending from the oral cavity down through the esophagus, into the stomach, and across the intestines. Mapping this extraction process requires looking at the specific mechanical forces and localized chemical environments operating at each stage of the tract. Human survival relies on the sequential execution of these destruction phases. If any stage in the pipeline fails to perform its specific biomechanical or biochemical task, the organism cannot access the energy it needs. The oral cavity is the initiation point. Here, teeth apply raw mechanical force to crush food, shearing apart the macroscopic physical structure of the material. Simultaneously, the salivary glands secrete an enzyme called amylase. This begins the chemical cleavage of carbohydrates, splitting them into smaller units and binding the chewed material into a cohesive mass known as a bolus. The tongue forces the bolus into the throat, routing it to the flexible esophageal opening. At this point, the primary mechanism shifts from chemical cleavage to active mechanical propulsion. This diagram shows the tract's cross-section, revealing a two-part muscular engine, an outer longitudinal layer and inner circular fibers. Contracting in sequence, they create peristalsis, an alternating wave powerful enough to pump the bolus to the stomach even if standing upside down. The food is not falling, it is being actively pumped into a hazardous chemical environment. The bolus enters the stomach by passing through the esophageal sphincter. This acts as a one-way pressure valve that relaxes to let the bolus pass, then immediately contracts to prevent caustic contents from traveling back up. The stomach is built for intense physical stress. Its inner walls feature folds called rugae that allow the chamber to expand, while a third oblique muscle layer over the mucosa is designed specifically for intense mechanical churning. As the stomach stretches, or in response to sensory inputs like the smell of food, specialized gastric glands begin secreting 1 to 2 liters of gastric acid every day. This biochemical map illustrates hydrochloric acid's dual function. It destroys incoming bacteria and interacts with inactive pepsinogen molecules. The acid converts pepsinogen into pepsin, the active protease required to break down complex proteins in the stomach. The gastric glands also deploy gastric lipase, which initiates fat digestion by splitting triglycerides into free fatty acids and diglycerides. Crucially, these same glands produce intrinsic factor. While it performs no function in the stomach, this molecule is required later for the intestinal absorption of vitamin B12. After 20 seconds of violent, intense mechanical churning, the acidic mixture is pulverized, becoming a semi-fluid mass of partly digested food called chyme. The stomach survives holding this corrosive mixture only by constantly secreting a protective mucus layer that shields its own mucosal lining. Virtually no nutrients have been absorbed at this stage. The stomach operates as a decontamination and chemical preparatory chamber. The chyme is released through the pyloric sphincter at the bottom of the stomach. This exit valve selectively meters out tiny amounts of the acidic chyme into the first section of the small intestine, the duodenum. The pancreatic enzymes required for the next phase of digestion cannot function in this acidic environment. They would be instantly denatured by the stomach's low pH. Pancreatic juices flooded into the chamber contain sodium hydrogen carbonate, which rapidly neutralizes the acidic chyme to an optimal pH of 7 or 8. This happens at the ampulla of Vater, where pancreatic secretions and liver bile are injected. Once the alkaline environment is stabilized, the duodenum wall releases enterokinase, which activates the pancreatic proenzyme trypsinogen into trypsin. Trypsin then cleaves the remaining proteins and triggers other enzymes to follow suit. Simultaneously, hepatic bile from the gallbladder attacks the lipids. Through a process called emulsification, bile shatters large fat masses into thousands of microscopic droplets. By pulverizing the fat, bile increases the total surface area exposed to pancreatic lipase, allowing it to cleave the triglycerides into free fatty acids. The duodenum operates as an environmental airlock, isolating the acidic chyme to unleash a calibrated alkaline enzymatic assault. As the chyme travels onward into the jejunum and the ileum, the pipeline's priority shifts from chemical breakdown to molecular absorption. To pull nutrients out of the fluid, the body relies on specific geometric configurations. The first level involves circular folds extending about 1 centimeter into the intestinal lumen, forcing the chyme into a convoluted path. The architecture scales down. Folds are covered by millimeter-long villi, multiplying absorptive surface area. Inside each is a dual transport system, red blood capillaries wrapping a central lymphatic lacteal. Simple nutrients transfer into the blood, while larger fats route through the lacteal. The final section of the small intestine, the ileum, lacks circular folds. Its role is scavenging remaining resources, sweeping up electrolytes like calcium, trace elements like zinc, and absorbing the vitamin B12 bound to intrinsic factor. Nutrient extraction at this scale is a problem of geometry, solved by compressing a massive, folded absorptive surface into a restricted biological space. Whatever cannot be absorbed is passed through Bowhin's valve, leaving the small intestine and entering the large intestine. The internal architecture here changes. There are no villi on the walls of the large intestine, as the primary extraction of digestible nutrients is complete. Instead, this environment is dominated by an estimated 100 billion resident bacteria. This microbiome performs tasks the host tissue cannot do alone. They decompose indigestible fiber to generate trace energy, synthesize essential vitamins, and neutralize competing germs as part of the immune system. Peristalsis drives the waste through the ascending, transverse, and descending colon. During this transit, the large intestine reabsorbs water back into the bloodstream and adds mucus to lubricate the remaining solid waste for excretion through the rectum. The large intestine functions as a symbiotic bioreactor and a water reclamation facility, extracting the final utility from the biological waste. In a matter of hours, organic material is mechanically crushed, sterilized in an acid bath, neutralized in an alkaline environment, stripped of its molecular value across nested surfaces, and dehydrated by a microbial colony. This is achieved through gated pH environments, the sequential activation of cascading enzymes, and extreme spatial optimization. The human digestive tract operates as a compartmentalized disassembly pipeline, an evolutionary architecture that extracts the fuel required to sustain the organism.
Thank you for watching. If you enjoyed this video, please like, comment, share, and subscribe to Neuromind Insights for more.